All right, so it's my pleasure to announce Dan Graur.
Yeah, I saw that.
I did a postdoc with him for 2 years before I came here.
I'm going on for
So I'm glad he's coming to talk to us. So Dan's originally from Romania, but he spent most of his youth in Israel, and that's also where he got his bachelor's and his master's. He got his PhD with Masatoshi Nei at the University of Texas, Houston, and then went to Germany for a postdoc, went back to Israel for a faculty position, and then in 2003 moved back to the United States, back to Houston to become John and Rebecca Morey's professor at University of Houston, where he basically rebuilt the ecology and evolution biology section of his department from the ground up. Very successful at that. Over his career, he's written over 125 papers. The important textbook.
This will come up in December.
In our field, so yeah, this is what.
Your task now is for each of you to buy 6 copies.
So important textbook. A lot of us, I know me personally, learned first about molecular evolution from his textbooks. He is a fan of fine art and Broadway, he and his wife. His house is like a museum where they buy art from Houston artists and put it sort of everywhere because his kids are grown up and they're not tearing things down anymore. One of my famous stories, so he's a fan of Broadway. He's very excited that one of his sons lives in New York. And
reason to visit.
And so we were going to a conference in Dallas. So of course I was driving the rental van with the lab in the back and Dan's riding shotgun. So halfway through the trip he pulls out his laptop and he starts playing Broadway songs on the laptop driving and telling us what city and when he saw the musical, whether it was good or, in Dan's language, shit. And giving us his opinion while I was trying to drive and no one else cared, I think. But he was trying his best to give us some culture. And so I think now he's going to give us a talk and try best to give all of us some sort of scientific culture.
Thank you. Unfortunately, there was not a place here for me to bury myself, but okay, or to hide. Let's start with flying on United Airlines. And since I have never slept on any flight, I tend to read everything that is in there. And they have an Hemisphere magazine, and I saw this ad. It says, the origin of species, Seiko Astron, like a smartphone, it's GPS enabled, allowing it to determine accurate time, blah, blah. Unlike a smartphone, however, it looks nice with a suit, won't break if you drop it, and uses solar power, so it never needs to be charged. Darwin will be proud, $2,300. And then I realized that the evolution of the species and Darwin would be proud is how many people regard the evolutionary process, that it produces perfect stuff. And actually it doesn't. So what I would like to do now is talk a little bit about my take on evolution, what is evolution, and whether it produces perfection of any kind. So first of all, as opposed to people, as you probably know now, evolutionary biology is done by people who have never taken a class in evolution. They are called computer jocks, MDs, and other kinds of people. So evolutionary biology is a mature science. It's a coherent discipline with a handful of logical principles, each of which has repeatedly withstood rigorous empirical and observational function. And evolution is merely a change in allele frequency. There is nothing more to it. C'est tout, like they say in French. Evolution process does not have to be either gradual or progressive. So anything is that. The other thing is that the rules of evolutionary biology apply to all levels of study. Whether you study DNA or a genome or morphology, the same rules apply. It's just the difference of evolution. In fact, that's how I end up my introduction to the new book. Understanding a handful of principles is all there is to evolutionary biology. The rest is details. And the details, unfortunately, in my book take about 950 pages. So first of all, evolution does not give rise to perfection or anything resembling intelligent design. I would like to see a wheel on a fixed axle in evolution. It hasn't produced it yet. So evolution does not produce better, fairer, more efficient, more honest, cleverer state. It just produces changes that are not excessively deleterious. And what can we say about the products of evolution is that most of the products work, some efficiently, some only barely, and some in a method that is quite clumsy. The example I use is, this is an intelligently designed dining table. This is a dining table designed by evolution. Take something that exists, turn it on its side, and it will work perfectly as a dining table. And if you don't believe me that evolution doesn't produce perfect stuff, just look at the evolution of the vas deferens in humans, and then you will see that whoever designed it could have used a good GPS. Now, if genes, genomes, and organisms were created by a benevolent, intelligent designer, the problem of evil would never have arisen, and also Department of Pediatric Oncology will never be needed. So the other thing that many of my students have problems with is to understand that evolution does not occur at the level of the individual. The only thing an individual can do is either produce offspring or die childless. That's the only thing he can do. And the only things that can produce an evolutionary process is selection and random genetic drift. There is nothing else. And because populations are finite, all the biological populations are finite, change always occurs. So unfortunately, the change doesn't create, in the language of Voltaire, the best of all possible worlds. So the best of possible worlds usually isn't. This is NA student number 8. I'm NA student number 7. We are numbered. I noticed that you are bringing here, or brought already. I saw Makova here, so she's a much higher number, which means that she's younger. How well selection works depends on effective population size. And effective population size is an historical construct. Census population size is just a snapshot of the present. And selection is not very good in populations with small effective population size. So if you take humans, there are about 7 billion of us in the world today. However, the effective population size, the long-term effective population size, is about 10,000. On the other hand, if you take chimpanzees, and they're numbered in the hundreds of thousands, and that's a very generous estimate, their effective population size is about 10 times that of humans. So the fact that there are a lot of us today, it doesn't matter much. So humans have a very small effective population size, which means that selection is not very effective, and you can look around you. I mean, we haven't evolved much. The other thing is how change occurs in evolution. During evolution, it's much easier to create equivalence than improvement, and it's much easier to destroy a function than to create a function. Again, it's the same with windows. It's very easy to break windows. It's very difficult to do it. Now, another thing that many of the, how should I call it, badly trained technicians with lots of money don't understand is that humans do not occupy a privileged position in evolution. Our genome is smaller than that of onions. We have fewer genes than cassava. The human biology is really very boring if you think about ciliates. Human populations are smaller than tardigrades, and human genetic variability is nothing in comparison to C. elegans and C. brenneri. In fact, that's something that ever since Copernicus, the anthropocentric worldview has repeatedly been shown to be erroneous, and yet the people who have the money continue to spend billions of dollars in search of human genomic perfection, producing along the way a great many headlines and precious little science. One of the ways, by the way, with which you can detect crooks in science is if they proclaim that what they do is a paradigm shift and the textbooks need to be rewritten. Okay, so my personal story starts on September 6, 2012. Until then, I was a very nice person. However, on this day, the ENCODE project consortium produced this sentence. This data enable us to assign functions to 80% of the genome. And in fact, on the same day in Science, the ENCODE project writes eulogy for junk DNA. So you have to see that it's a very impressive study. It's 147 cell types, 442 researchers, $288 million. Think about it. It must be much more important than deciphering the structure of DNA, in which 2 people were involved and it cost nothing. So what happens on this day is that this is how I was before that. Laptop, water, coffee, and this what happens later. And in about 2 years, I have morphed into the vigilante. So everybody now knows that I cannot stand squirrels. And the reason is that because they are like the people who manage ENCODE, they are rats with good PR. So what is a biological function? That's what I will deal today. The nomenclature of genomic function, the evidence for the existence of vast quantities of junk DNA, the mutational mechanism responsible for generating junk DNA, the conditions for maintaining junk DNA. And one of the things that I will deal towards the end, I'm not going to deal with all these questions, is what went wrong, how these people got everything wrong. So let's start with what is a biological function. Biologists mostly use what's called a historical concept of function, or the selected effect function. But there is another type of function which is called the causal function. So if something has a selected effect function, you have a trait that performs a function, and the trait originated as a reproduction, a copy of some prior entities that performed this function or a similar function in the past. And A exists because of this function. So essentially, a selected effect function answers the question, why does T exist? Why does a trait exist? On the other hand, a causal function, it's just what does T do? Just to give you a simple example, the function of the heart. If you go to the selected effect function, the function of the heart is to pump blood. But you have other causal functions of the heart. It adds 300 grams to the body. It produces sounds. It provides an inaccurate logo for Valentine's Day, and also pumps blood. So every selected effect function is also a causal function, but not every causal function is an effective selection function. And most biologists use the selected effect concept, but badly trained technicians with too much money use the causal function concept. So just to give you one example, from many papers, operationally we define a functional element as a discrete genome segment that displays a reproducible biochemical signature, for example, protein binding. So do you know what is the function of my shoes? The function of my shoes is in Houston to bind chewing gum. Houston is a very hot place, and people throw things on the sidewalks. So this is a function that fits the definition that was used by some of these people. Unfortunately for me, another person, Lawrence Hurst, had a much better metaphor for that. By the logic employed by ENCODE, following a collision between a car and a pedestrian, a car's bonnet, I'm sorry, he's British, would be ascribed the function of projecting a pedestrian many meters, and the pedestrian would have the function of deforming the car's bonnet. So this is a function. I mean, that's what pedestrians are made for. It seemed to me that what was needed was some nomenclature of what genetic function is, and especially as far as junk DNA is concerned. Now, these are a typical example. All of these three people are MDs, and this is how they define function, junk DNA. In genetics, the term junk DNA refers to regions of DNA that are non-coding. What bullshit, as if molecular evolutionists never knew about tRNA or something. The same thing, that's how they are. The genomic regions that encode proteins, the genes swim in a sea of non-protein coding sequences or junk DNA. So, there needed to be some order into the nomenclature. So let's first look at what the genome does. Some genome is not transcribed, some is transcribed. Of the part that is transcribed, some is translated and some is not. In all of these cases, you have functional parts and rubbish parts, things that don't do anything. So you can have rubbish proteins, rubbish RNA, and rubbish DNA. And in terms of function, the reason I used rubbish is because I adopted the nomenclature from Sidney Brenner, one of the most brilliant people that ever existed in my profession. So I divide the genome into a functional part and the rubbish part. And the functional part is very simple. Everything that has a selected effect function is functional. Everything that doesn't is rubbish. And I'll sit there. I'll die of heat. But I'm connected to wires, and I'm not very good at hardware. So let's look. This is a paper we published this year. So let's look at the functional DNA. There are two types of functional DNA. There is literal DNA and indifferent DNA. And literal DNA is essentially any piece of DNA in which the order of nucleotides or some order of nucleotides is under selection. For example, there are 3 possible sequences of length 3 that can encode isoleucine according to the standard genetic code, as opposed to about 64 possible 3 nucleotide sequences. So the reading of it, despite the fact there is some ambiguity in it, the order of nucleotides is important. So this is literal DNA. Indifferent DNA is sequences that need to be there, but the content and the order of nucleotide is not important. Probably the trivial example of indifferent DNA will be the 3rd position of 4-fold coding codons. It doesn't matter if you have a T, C, A, or G, but if you don't have it, you will have a frame shift. Now, rubbish DNA, the entire nomenclature here is taken from Sidney Brenner, and he writes so well that I don't have to do anything but read what he wrote. Some years ago, I noticed that there are 2 kinds of rubbish in the world, and that most languages have different words to distinguish them. There is the rubbish we keep, which is junk, and there is the rubbish we throw away, which is garbage. The excess DNA in our genomes is junk, and it is there because it is harmless as well as being useless, and because the molecular processes generating extra DNA outpace those getting rid of it. Now, the continuation has some sexist things, so I don't know, in modern universities you have to give a warning. Where the extra DNA to become disadvantageous, it would become subject to selection, just as junk that takes up too much space or is beginning to smell is instantly converted to garbage but by one's wife, that excellent Darwinian instruments. So, here you have a good example of what is what. This is my garage at home. I can park my car, but it's full of junk. This photo that I took from the internet is no longer functional. The junk became garbage. It can no longer perform function. ENCODE, for example, thinks that all houses should have this. Anyone knows what is this picture taken from? That's the collection of cars of Jay Leno. And that's not your typical genome. Okay, now genomes also evolve. you can take the four compartments, the literal and the indifferent and the junk and the garbage, and they can transform one into the other. Probably the easiest transformation is to take functional and turn it into junk. That's how we create pseudogenes. In fact, one of the pseudogenes in humans that drive me mad is the fact that we are unable, humans and guinea pigs and all primates actually and all hystricomorphs, are unable to synthesize ascorbic acid. And because we are unable to do that, we have to eat lettuce. And lettuce, according to my zoology professor, is not food, is what food eats. And the reason is that we cannot produce ascorbic acid like our dogs and our cats and our cows is because one enzyme on the pathway from glucose to vitamin C died. And because it had no functional copy in the genome, we have scurvy. And we have to eat horrible stuff. Which, of course, according to the paleo diet, you should not eat. Why shouldn't we adopt a diet of a period in which the average lifespan was about 16? Of course, junk can become functional too. I call it Lazarus DNA. I use Lazarus because it's the second most famous resurrection in fiction. And junk can be repurposed. But we know about many cases, and probably the most important cases are for what's called domesticated transposable elements. Sometimes, however, junk may become garbage. Something that used to sit there and do nothing suddenly activates a splicing site or something, and suddenly it causes problems. And this is called zombie DNA. This name, by the way, is due to Gina Colata from the New York Times. For example, our nuclear genome is full of sequences derived from the mitochondria. They are called numides. And of course, you have allo elements, but some allo elements can enter into an intron and then activate the splicing site, and then instead of getting a good protein, you get something very bad. What is the evidence for existence of vast quantities of junk DNA in the human genome? First of all, we have what is used to be called genomic paradoxes, a lack of correspondence between a measure of genome size or genome content and the presumed amount of genetic information needed by the organism. And I will not go into the difficulties in measuring complexity. Complexity is used not only in biology, but it's used in engineering and things like that. So in engineering, for example, complexity is the number of variables that are needed to describe an object, which is not a very good measure in biology because a cracked wall will be more complex than a wall that is not cracked. But in many cases, although you cannot define complexity, you know for certain that some organisms one is more complex than other. A unicellular dinoflagellate, for example, is probably less complex than this guy. I suppose it's your president, your governor or something? I took something from Arizona. Who is he? Governor, okay. No, I didn't know anything about your president. I didn't want to insult him. In Arizona, however, you know for certain that he's a Republican, so you can insult. And this dinoflagellate has a genome that is 28 times bigger. Of course, this is a subjective genomic paradox because we always think that humans are more complex and they are the pinnacle of creation. However, you can turn these paradoxes into objective ones. we can look, for example, at the number of cells, or the number of cell types, or the number of cell tissues. And we can get numerous slices of evidence that there is no relationship between genome size or genome content and organism complexity. And the two most famous paradoxes, one is C-value and the other one is G-value. If you look at the C-value paradox, we are here. The famous onion is 5 times bigger than us, but you also have unicellular organisms that have more higher genome sizes than we have. But also, notice that if you take 2 species belonging to genus Paramecium, it's a huge differences in genomes. So this is the C-value paradox. This is currently the organism with the highest haploid genome size. It's called Paris japonica. It doesn't even have a good colloquial name. And this is what Ryan Gregory suggested in 2007, the onion test. Is a simple reality check for anyone who thinks they have come up with universal function for 80% of the genome or 100% of genome. Whenever whatever the proposed function, asks yourself this question: can you explain why onion need about 5 times more DNA than humans? There is one possibility, however, to explain it, and that is assuming that the humans are the Goldilocks of genome size. So you probably, I don't know how many of you remember Goldilocks. It cannot be too small or too big. It has to be just right. Now, let's assume that humans have no junk DNA. It's all functional. So, but you cannot assume that onions have more genes than we do, or they are more complex, or more the pinnacle of creation. So we are the only ones who do not have junk DNA. Everything else will have junk DNA. So that's how we become the Goldilocks. And the G-value paradox is another nice story. It's about the number of genes. Okay, since I'm old and I collected posters at one time, NIH used to produce posters. And like everything that is bureaucratic, it's essentially a form. This poster is YGA 98090R. I got it in 2000. Now I notice that this poster is from 1998. And it tells students something about the DNA. We have trillions of cells. Each cell has 46 human chromosomes, 2 meters of DNA, blah, blah, blah. And the genome encodes 80,000 genes that perform all life function. And then, of course, in 2001, the human genome is out. So they have to produce another form. And the change is that suddenly we don't have 80, we have approximately, and that perform most life functions. So please notice, and what happens since is really horrible. Really, really horrible. The first person to estimate the number of genes in the human genome, it was done in 1964. It was 6,700,000 genes. No, really, that was the number. It was published in Nature, and everything that is published in Nature is correct. In 1990, the number went down to 100,000. About here, we start having data. And currently, we believe that there are 19,000 genomes. Why do I have it twice? I don't know. This is the newest number that I have is a paper by Escordia et al. That claims that we have as few as 19,000 human protein coding genes. There is a phenomenon in annotation, because people who annotate genome know that there are errors in annotation, and that's called the Mad Magazine version of the human genome. You take pseudogenes and you resurrect them in silico. So you assume that this deletion here is probably an artifact, and you resurrect gene. And this is the other paradox, the G-value paradox. We have about 2,000 genes less than C. elegans. And currently, the triticum is the bread wheat is the. So that's how I imagine every human newborn starts screaming because the fern has 27 times as many chromosomes as I do, and the stupid flower in Japan has 50 times more DNA than I do, and wheat has at least 5 times more genes than me. And it's quite annoying, but of course everything can be corrected by public relations. So, is your genome big? Oh, it's amazingly big. This is what the NIH tells you. That the total length of DNA in an adult human, they have an educational site that you should never use unless you want to educate ignoramuses. The total length of DNA of an adult human is 2 times 10 to the power of 13, the equivalent of 70 trips from Earth to the Sun and back, or 5 trips from the Sun to Neptune and back. I was always wonder why Neptune and not Uranus, but maybe because of the language. But actually, I can show you that your genome is really not that bad, not that big. It has 3.5 billion letters in a 4-letter alphabet, which is about 7 billion bytes, or 0.8 gigabytes. And so, my God, in a DVD, you have about 10 times more information than in the human genome, which makes this movie the paragon of all creation. Or you take all these very important people that you may recognize, and you add a C. elegans in it. Now, many years ago, people don't really understand, but some people did science in the 1930s and 40s and 50s and 60s and so on. Science did not start yesterday. People were looking at the question, is it even possible to create a genome that is entirely functional? And the way they did it, and I'm not going to teach you a genetic load now, but the way they did it is that the fitness of an organism is never the best fitness. And the reason is that deleterious mutation may occur faster than selection can get rid of them. Recombination may break apart favorable combination of alleles. And genetic drift may cause allele frequencies to be suboptimal. And you can do all the calculation of genetic load and connect it to mean fertility. Again, I will not do that. When I do it in class, it takes me forever. But you can look at that, and under these assumptions, that the size of the entire haploid human genome is 3 billion, the entire genome is functional, the length of each functional locus is 1,000 nucleotides, the mutation rate is 10 to the minus. There should be a minus here, I'm sorry. And most mutations are deleterious. If you do that, then you realize that each of us has to have this number of children in order for one of them to be alive. And that in human population, I don't know. I know that some people don't use contraception methods, but I have never seen anyone with this number of. So the only solution, and that's how Motoo Kimura came to the neutral theory, is to assume that most mutations are neutral. And the reason they are neutral, because they occur in genomic regions that are devoid of function. What are the mutational mechanisms that are responsible for originating functionless DNA and maintaining functional DNA? We now know the answer. Most of the junk DNA that we have and other organisms have is derived from transposable elements. Mostly they're incapacitated descendants. The minimum number currently in the human genome, it is assumed that about 60% of the junk DNA is due to transposable elements. Of course, this is a minimal estimate because if something doesn't have a function, it will evolve very fast. It will change its sequence and may not be recognizable as a transposable element. At present, I think they found only 2 yeast species with very small genomes that do not have transposable elements. But in some huge, currently the biggest sequence, the biggest genome ever sequenced belongs to this loblolly pine. I have no idea what it is, but it's a pine, I suppose. This one has a huge genome, and most of it is transposable elements. I mean, by most of it, I mean 95% of it. How do you explain the accumulation of so much junk DNA? The most plausible hypothesis, in my opinion, is due to Lynch. He assumed that having a big genome is not good for you. So each addition to the genome will decrease a little your fitness. However, because in population with small effective population sizes, if the effective population size is small, then this little decrease in fitness will not matter. And the key prediction from his theory is that if you compare effective population size to the genome size, you will find a negative correlation. So this is a measure of effective population size. I will not tell you what this measure is, but there are different measures. They are proxies because it's very difficult to count directly effective population size. What do I take the entire human population, throw out everybody who is related, throw out the nuns, throw out the people who ride motorcycle because they will never reproduce, and so on and so forth? No, it's very difficult. But we can use measures of heterozygosity and measure of variability to get some measure of effective population size. And we see a very nice correlation with genome size. Organisms that have very small effective population sizes have very large genome. You seem to be very unhappy with that. You want to have a smaller genome and more effective one. I know. You will, by the way, when you get tenure. So how do you estimate the functional function within genome? It's very easy. You ask yourself, is it possible to destroy the function of this thing? Is it possible to destroy the function of this thing? No. No, I mean, you can beat it up. I mean, what else? It will achieve less maximum velocity? No, that's... so one of the things that happen in evolution is that if you have selection, many mutations will be thrown out. They will be deleterious. It is possible to damage a functional car. But if it doesn't have a function, you don't have a sieve anymore. So every mutation has an equal chance of becoming a substitution in the population. So actually what you have to do is essentially, and I notice that some people do it here, they look at the rate of evolution, the rate of substitution, by comparing different organisms. Another measure of functionality, of non-functionality, is asking yourself, what happens if I delete this part? And some people did do it. They did mega-base deletions of gene desert. Gene desert are regions that have no known genes there, and they result in viable mice, and they are happily ever after. When they published this paper, these mice were at generation 13. Now there are many more, and Ed Rubin tells me they are still alive and well. Of course, what happens if you freeze them, if you don't feed them, if you, it's very difficult to cover all possibilities. But one thing you have to remember, in science, your hypothesis should specify the conditions for their rejection. So if I say that something has no function and you do find the function, then you can reject my hypothesis. However, if you claim that a certain area has a function and you search and search and search and you have no function, you said you have searched enough. or under the right conditions. So actually claiming that something has a function on the basis of very flimsy evidence, because in one molecule out of 220,000 samples that you have, you find that it binded a transcription factor. That's not a good evidence that it has a function. So what fraction of the genome is functional? I don't have to rely on myself. In 2014, Rand, Sponting, and Lanter conducted probably one of the most thorough studies on conservation in genomes, and very good statistical thing. I happen to be one of the reviewers of this paper, and when the first paper that they wrote had a different title, and because they know on which side the bread is buttered, they said, 8.2% of the genome is functional, and it agrees completely with the results of ENCODE. And I asked him exactly which universe 8.2% is equal 80%. And I forced them to change the title. That's what happens when you edit stuff. You can do things, especially if the people are younger than you and want tenure. So actually what we know now is that 90% of the human genome is junk. Now, you ask yourself, how these people, I mean, we know that it's 8% functional. How did they claim that it's more than 80%? One thing, they claim that a biological function can be maintained without selection, which in turn implies that no deleterious mutation can occur. And that's why it gave us the idea for the title of the paper. This is akin to claiming that the television set left on and unattended will still be working condition after 1 million years. We submitted it, originally the paper, to Trends in Genetics. They refused to publish it unless we remove invective, sarcasm, vituperation, polemics, ridicule, zingers, and slander, which would have left a very, very small article, and we refused to do it. So this is a short list of what went wrong with ENCODE. I will just want to give you a few highlights. They used cell lines. The vast majority of cell lines used in research are misidentified. They used HeLa cells. This is the karyotype of the HeLa cells. Do you recognize the human karyotype here? I don't. In fact, in 1991, Van Valen and Majorama defined HeLa as a new eukaryotic microbial species. It's a parasite. It lives on people with white coats, but it's still an organism, and it doesn't look like human anymore. The other thing that I like a lot is post-mortem tissues. Now, if you use a lab animal, like a rat or a mouse or Drosophila, you can use tissues very easily because you can obtain all the tissues at the same time. But unfortunately, the NIH does not allow you to flash freeze humans when they are alive and were well. So they use post-mortem tissues. And nobody takes into account the impact of RNA degradation on transcript quantification. This is a new paper. I'm always suspicious when you use gene expression in dead tissue from long dead elderly humans. And I think it's like studying blood circulation in sushi or mating behavior of roadkill. I just don't like it. Psychological errors. Humans are not very good at dealing with randomness. In fact, I had a very nice conversation with Dr. Nishi, and he said that making our mind very quickly, seeing patterns in what is otherwise random stuff, may have an evolutionary advantage, may have a selective advantage. That may be so in evolution, but scientists should be above it. By the way, there is There is a term, by the way, for people or for this affliction where people see meaningful patterns or connections in random or meaningless data. It's called apophenia. You may have probably seen newspaper articles in which people see Mary, the Mother of God, in a toast, piece of toasted bread and things like that. My best example is this book, The Bible Code. I can tell you anything I like. I don't know how many of you can read the Hebrew letters. But what they do in the Bible Code, they take the Bible and they write the letters, I'm sorry, from right to left, in matrices. And then if you find things like here or here, you know that this is something very important. So let me show you one example here. Mitt Romney Nasi, president. As we know, this was good, no? These are what's called the Torah code. They are still being used, by the way. You can see them all over the internet. The other thing is the word junk. Junk has a semantic baggage. People don't like to use it. I should have given you a warning. Yeah, I told you that a week before I came here, there was an official complaint logged against me because I used the D word. It took me some time. I used the word damn. So, logical errors. One of the most horrible logical errors committed by ENCODE was affirming the consequent. So if something performs a function and has a certain property, if something else has the same property, it most probably does the same thing. So we know that some functional regions are transcribed, ergo all transcribed regions are functional, and so on and so forth. Now, how do they reach the 80%? This is an amazing thing. They did all this. Genome is transcribed, associated with modified histone, found in open chromatin areas by transcription factor, consists of methylated CPGs. So they took essentially the union of all these things and found out that 80% of the genome is functional. Actually, I think the 100% of the genome is functional. 100% of the genome is replicated. So at least it attaches DNA polymerase. So it's functional. Statistical errors. Now, one of the most annoying things in the new science is that we have trillions and zillions and gazillions of data. Now, when you have so many data points and you conduct so many statistical tests, statistical significance has absolutely no meaning. If 5% of the trillion experiments that you did, statistical experiments that you did, will be statistically significant under the definition of 5%, which is itself arbitrary, there is nothing you can do with it. So let me give you just one example. You can see that, I mean, there are certain journals, for example, like Nature Genetics, for example, it likes to publish things like that. Common genetic variants associated with cognitive performance identified using proxy phenotype method, which essentially will be translated in the newspaper, IQ is heritable. There is nothing you can do. Don't put money into public schools because you cannot improve it. Statistical significance, 5 times 10 to the minus 8. What can be more significant than that? And it's all written in the abstract. Unfortunately, what is not written in the abstract is how much of the variation is explained by these 69 SNPs from 106, 736 genomes. It explains 0.02%, which you should add 2 more zeros, of the variation. So like my supervisor for the master's degree says, oh, it's statistically significant. Is it even interesting? So you can use methodologies that encourage biased errors. You can consistently favor sensitivity over specificity. You should pay attention to significance rather than the magnitude of the effect, and so on and so forth. One of the things that people did not pay attention to, or don't do it very well, is batch effects. We know that every experiment that is done is affected by the person who makes the experiment, the temperature in the room, how many people are there. So what should be done in order to avoid batch effect is that experiments should be set up so that batches do not overlap with biological variables. So if you have one experiment to do, you should give part of it to be done in Luxembourg, part of it in Texas, part of it in Arizona. But that's not how it's done, because labs are not built that way. So everything that has to do with one experiment is done in one lab, and the other experiment is done in another lab. And what the people, like the people in ENCODE, want to tell you is that all the samples were dealt with according to exacting uniform criteria. What does exacting uniform criteria means? So there are about 100 institutions in the USA, UK, Spain, Switzerland, Canada, Brazil, Netherlands, Russia, and Estonia. And they want you to tell me that all these labs conducted it according to exacting uniform criteria. And if you believe that, I suggest you put a bid. I have a copper statue for sale on eBay. And then you have something that makes me very angry personally. And the reason is that big science costs a lot of money, and then there is no money for real science. I'm sorry. This is about 1,000 normal grants that have not been funded because of that. So is big science worth the cost? So you take ENCODE and mouse ENCODE and GTEx and Phantom and Blueprint. They're very good, by the way, at acronyms. One thing that I noticed in most of this article is that they start and end the same. We use a very large collection of something, and at the end of it, we look forward to scaling up the resource. This reminded me of a person by the name of Charles Ponzi. So what do I think about such studies? I can quote no better scientist than Frank Zappa. Data is not information, information is not knowledge, knowledge is not wisdom, wisdom is not truth. So, a better use for money, by the way, here. Thank you so much.
Questions? Sure.
And a lot of computer programs from Microsoft and so forth, they build upon previous programs. And so we have today that a lot of these programs, they have no idea what the subroutines do, and it's a mess, but they can't redo it, so they just know some of the functionality.
My current problem in my lab now, we started working on parallel and convergent mutation. Doesn't matter when the same things happen on different evolutionary lineages. And in order to do that, we used two methods of reconstruction. One is maximum parsimony, one is maximum likelihood. Theory says that maximum parsimony should yield the least number of changes, because that's what maximum parsimony is. It looks for it. But actually, it doesn't happen that way. In fact, there are cases in which maximum likelihood yields a less number. So now, instead of doing the research I intended to do, I have to look into the bowels of the program, and I'm not very good at that, to look what happens. And I cannot, of course, talk to the person who wrote Mega, because he is not on speaking terms with me. That's the only reason I came here, because you got rid of him. Sorry. And instead, he brought a very nice person that I met in Barcelona a few years ago, yes. Yeah, it's like that. Actually, the metaphor of the computer program is very similar to the metaphor of genomic evolution. Because in the evolution of programs, of course, it's not really evolution. the word itself is used as a metaphor, you end up with what's called dead code. These are parts of the code that can never be reached by any routine, but they are left there, and nobody dares touch them, because probably, although it is never reached, probably the moment you remove them, you will cause a big problem. So yeah.
Anyone else? All right. Dan.
Thank you.